All right, hi everybody, and thank you so much for taking time out of your afternoons to come to our workshop on validating antibodies for better science and better medicine. Your speakers today are going to be all from Fortis Life Sciences. So I'm Aaliyah Weinstein. I'm a Senior Marketing Programs Manager at Fortis. And throughout the talk today, you're going to hear from my colleagues Brian McWilliams, who's a Senior Product Manager, and Amber Miller, who's our flow cytometry scientist, talking to you about the antibody validation methods that we use at Bethyl Labs, which is a Fortis Life Sciences brand. Our brands at Fortis support your entire R&D pipeline by providing comprehensive solutions that power scientific discoveries from R&D through manufacturing. So Fortis is an umbrella company, and we have several brands underneath that umbrella that offer CDMO and CRO services across a variety of therapeutics and diagnostics categories, as well as catalog reagents and custom antibodies for a wide variety of immunoassays that you might be using in your own research, including IF, IHC, flow cytometry, Western blot, ELISA, and more. We have 3 brands that provide antibody-based reagents, which are Arista Biologicals, Abcor, and Bethyl Laboratories. And today you're going to be hearing from the team from Bethyl. Bethyl has been making and validating your antibodies for over 50 years, and our catalog includes over 6,500 primary antibodies, over 1,200 secondary antibodies, over 50 ELISA kits, 50 antigen proteins, and more. And our antibodies have been cited over 40,000 times in the literature. If you can't find what you need from that catalog, we also offer custom services, including custom monoclonal and polyclonal antibody development, custom conjugation of any antibody in our catalog, especially for use in flow cytometry and multiplex immunofluorescence, and newly launched just last month, our spatial biology CRO services, where we can support imaging by IHC and multiplex immunofluorescence all the way from tissue blocks to slide scanning. So I'm going to pass this off to Amber to talk more about our validation methods.
Before we get into the meat of validation, we wanted to bring up and remind you that back between 2002 and 2012, Amgen and Bayer did some extensive research and studies trying to replicate experiments that were done and published in the literature. And they were only able to replicate 30 out of 120 findings that were published in the literature. And the antibody companies kind of got the short end of the stick and were placed under blame a little bit in terms of why some of these replicates were not, or why there wasn't as much reproducibility. And for fair value, because at the same time, a lot of these studies were coming out where the antibodies were cross-reactive with different markers and different things that they weren't supposed to be. So like EPOR1 has 2 antibody clones that are cross-reactive with heat shock protein 70. You can see one at the bottom there, your C20 antibody here, and then this band for HSP 70. Plus a variety of other markers, right, that had an impact, right, on therapeutic and clinical biomarkers. Additionally, things like this were coming out in terms of how reliable are G protein coupled receptor antibodies, where out of 49 antisera to 19 distinct G protein coupled receptors, none of them were found to be specific for only one G protein coupled receptor. And things like this, right, where these investigators were, well, they spent over 2 years and over $500,000 thinking they were studying CUS-D1, when in fact they were actually studying CA125, which was not of interest to them. So because of this and the fact that billions of dollars are spent on commercial antibodies every year, right, you can see and easily imagine the impact that these poorly characterized antibodies can have on not just basic science, but translational research and clinical research. And because of this, a lot of information, a lot of studies have been done to increase our validation principles for antibody validation. But before we get into those pillars, what do we mean when we say validation? So according to the FDA, the process of demonstrating through the use of specific laboratory investigations that the performance characteristics of an analytical method are suitable for its intended analytical use. So in terms of the antibody, this is it reproducible? Is it specific to the target of interest? And then for your intended analytical use, is it validated for the different applications that you're actually going to use it for?
So at that point, the question kind of becomes a little bit more practical in how do we collectively, as both manufacturers and users of these antibodies, validate these products in a way that's useful and scientifically reliable for you. So in 2016, there was a publication that came out by the International Working Group for Antibody Validation that laid out sort of a set of scientific parameters that defined a series of what they called pillars that we could sort of collectively use to help validate these products in a way that could bring confidence to both to us as the manufacturer and you as the final end user. Now we, as a manufacturer, took this concept and sort of retroactively applied it to what we were already doing and doing very well, and adapted it to what we could do better going forward. So we actually used this as a guideline and then generated our own 6-pillar concept that we have illustrated here. And I will go over these in depth here over the next few slides. But we do have, of these 6 pillars, the first of them is the independent antibodies pillar, the complementary assays pillar, the orthogonal characteristics pillar, the biological characteristics pillar, the recombinant protein strategies pillar, and the genetic strategies pillar. And as I said, we will be going through these in depth over the next few slides. But whenever we're moving these things through our validation pipeline in-house, we require that 2 of these 6 pillars be achieved with each antibody that leaves our facilities. We have a subset of antibodies that have actually gotten up into the 4s and 5s. At some point after 2 or 3, you start to lose some value and just kind of lumping more stuff on top. So 2 is really our guideline, but everything that leaves our labs and our facilities has 2 of these 6 boxes checked off. So the first of these pillars is the independent antibodies pillar, and this is the one that we've really, I think, leveraged well historically. So even dating back to the mid-90s, we were using this concept to validate everything that came out of our polyclonal product line. And the idea here is that you're going to be using 2 antibodies raised against distinct epitopes of the same target protein. So you're ensuring at that point that if you're getting the same result from 2 distinct antibodies, you can trust that at a depth and a level that you cannot trust a single product. And so what we'll actually do, whenever we go after a certain target, we're going to actually use 3 or 4 different peptides in 3 or 4 different rabbits, purify those, and then we're going to perform IPs with each of those 4 antibodies, and then we're going to blot each of the 4 antibodies with each of the 4 products. And so you can see here in the bottom left, this is a experiment that we call a reciprocal IP. And so what we're doing here is we've got IPs generated from 2 distinct antibodies that are being Western blotted by a single one of these antibodies. And we can see here that despite these being 2 distinct antibodies against 2 epitopes, we're seeing the same target band. Mathematically, the probability of this happening on the same off-target protein is functionally 0. So this gives us an enormous amount of confidence that what we are seeing is actually what we are telling you that it is. We can also apply that independent antibody strategy in an experiment like this, where we have subsequent sections of the same tissue side by side being stained by these 2 antibodies that we've made on site, and we're seeing the exact same staining pattern. So this also gives us confidence. Conceptually, it's kind of the same, but it's not applied in quite the same way here. So the other half of that reciprocal IP is also the complementary assays pillar. In that case, we're using both an IP and a Western blot to provide us complementary results. In this example that we're showing you here, we're actually using a Western blot and an ICC assay to stain the same cell line that I have boxed in red here, SKML28, and we're seeing very strong reactivity in this cell line here and comparable reactivity there in the ICC as well. Moving on to pillar 3, this is the orthogonal characteristics, where in pillar 2, we're using antibody-dependent techniques to generate comparable results. In pillar 3, we're using 1 antibody-dependent technique and 1 antibody-independent technique to provide comparable results as well. So you can see here in the bottom left-hand corner, we've got data from Gene Visible, a publicly available database, and we see here boxed in on the bottom, both Raji B cells and Dowdy B cells are indicated as 2 of a small subset that show significant expression from the transcriptome. And we can see over here on the far right, the Western blot from those same 2 cell lines is very specific and giving us a nice clean band for CD19. So this is, again, a third method that we can use to show that the antibodies that we're generating are in fact on target. The 4th pillar that we use here is called the biological characteristics pillar, and this is where we are able to take advantage of the specific biology of a given protein maybe in a case where you have to induce expression, or we can phosphorylate that protein under a certain condition. So you can see here on the left under hydroxyurea treatment that we're inducing phosphorylation of CDK1 on residue 15. And over here on the right, we're able to induce phosphorylation of CAP1 using etoposide. We've got total CAP1 here, phospho-specific CAP1 here. We're seeing a nice strong pull down under both conditions when we're blotting with the phospho. This thing is on target. So again, this is a way for us to manipulate the biology of the protein to help us validate our antibody. Here in slide pillar 5, this is the recombinant expression strategy, and we will never ever only do this in a given experiment except in a very unique case like this. So in this case, we were looking at designing a MAF-A antibody, and we wanted to ensure that MAF-A was specific for MAF-A and specifically not binding to MAF-B or CMAF. So we created overexpressing cell lines for all 3 proteins, and then we're able to show our MAF-A antibody was only binding MAF-A, our MAF-B antibody was only binding MAF-B, our CMAF antibody was only binding CMAF, and then we have a MIC-tag control here in this center showing that, hey, there are cells in those lanes, we're just not seeing it with all of our antibodies. So this gave us a lot of confidence that we are specific for MAF-A and not seeing the other very closely homologous proteins. And finally, in pillar 6, this is the only pillar we will occasionally consider this as the sort of independent player, where if we can show a knockout cell line and we're showing specificity under that condition, we are going to be very confident that what you're seeing is what it is. So we have a ubiquinol 4 knockout here. We're seeing very strong expression in the wild type cell line. We're seeing no expression at all in the knockout, and we can see a very strong shift in flow here relative to our control. So this again gives us a ton of confidence that what's actually going into your hands is what we say it is. So functionally, like what does this mean for you guys, and what does this mean for us as a manufacturer? So you can see here that we have 3 very sort of distinctly kind of really beautiful over here on the left. We would call this an ideal circumstance or an ideal antibody, versus this over here on the right where it kind of looks ugly, but it's still really functional. So this is actually one of our best-selling antibodies, and you guys can see there's a lot of off-target noise down here, but during our validation process, we've over and over and over again across multiple rabbits, multiple lots, we've been able to show time and time again that this band here is MLL1. And we know that, the researchers who work on this know that, they buy this, it's very useful for them, but obviously it does not look as amazing as this nice clean single band here. So there is important value to recognize that things can be validated even though they're kind of ugly, and we try very hard to ensure that if you're going to buy a product like this, that you know what you're getting prior to that purchase, and that if you want to use that, it's valuable for you, but it may just not look great. So there are kind of a subset of challenges within the antibody space that our validation system is unique in allowing you to kind of figure out, like, hey, this is a problem, but because they've done it this way, we're confident that even though we have this issue, that it is still what we say it is. So in a circumstance where the predicted molecular weight and the relative molecular weight of a given protein is not the same, a nice clean band, like here on CAP1, we're seeing this thing migrating at like 100 or 110 kD. Well, the predicted weight's closer to 88. And so as an investigator, you might look at this and be like, well, this is a great band, but how do I know that that's CAP1? It's running too big. So because we've done a reciprocal IP where we have polyclonal antibody against CAP1, a different poly against a different epitope of CAP1, and then 2 lots of the same recombinant monoclonal against a different epitope of CAP1, and then being blotted by this antibody here, and we're seeing this nice clean banding pattern, and it's migrating right there about 100 to 110, we can translate these results here to this result here, and we can have confidence that this band here, even though its relative migration is a bit too high, is still what we say it is. We can also address issues of ubiquitous expression. So as you guys know, if a protein is being expressed at a high level across multiple cell lines, and you see a band in all these different cell lines, you don't really have a negative control there to give you confidence that, hey, this is what we say it is. So we're able to kind of use our pillar system to say, okay, well, let's use the orthogonal characteristics and say, hey, this is very highly expressed in NK92 cells. Let's run a reciprocal IP now with 2 different antibodies against different epitopes of the same target. We're seeing this nice clean banding pattern here. We know that that is what we say it is, okay? And this is also a particularly challenging situation where we have what appears to be a very distinct cell line expression pattern, but we're getting 2 weird banding patterns, okay? So we're seeing a single band here in jerkat cells and a 2-band pattern here in MCF7s. Well, my first instinct looking at this data is like, well, something's off target here. That can't be right? So instead of just relying on that, we're gonna fall back and we're gonna do reciprocal IPs now on both of those cell lines with 2 different antibodies against 2 different epitopes of the same target protein, and we're gonna show a distinct banding pattern now in both jerkat cells and the IP, which matches what we're seeing here, and that 2-band pattern in the MCF7 cells, which is what we're seeing here. And what was cool about this is that the literature actually confirmed that MCF7 does have this 2-band pattern when stained with this antibody, and this is due to a smaller isoform being expressed. So this gives us even further confidence that what we're seeing here across these 2 stains is the exact same as here, okay? And so with that, I'm gonna hand it back over to Amber to kind of walk you guys through the rest of it.
So up to this point, we've been talking about antibody validation in terms of reproducibility and specificity. We want to make sure that we're selling antibodies that are binding to the appropriate target. What we also love to do and what we pride ourselves on is validating for the specific applications that you're going to use these antibodies for. So you can get a lot of information from our website, from the data sheets, that's going to tell you what the applications that the antibodies are validated for. You also get information like species reactivity and things like that. We put all the information for basically the dilutions, the concentrations that you should use for these applications because we are testing them in-house and we are really validating them. So we're just going to walk through a few examples for these types of information. So Brian showed a lot of Western blot and IP data, and what's cool about this is that you're getting information about size that you can correlate with specific expression patterns. You can correlate this with the IPs. We also have a relatively new application validation for protein symbol, which is very similar to Western blot and IP, or Western blot, except it's run on a capillary system. So again, you can see our model system, our jerkets here. You can see increasing dilution, so you see a decrease in the intensity of the banding pattern here. So it gives you confidence, right, that you're seeing good things for this specific application. We validate for ELISA, and on the ELISA data sheet, you'll see stuff like this, where it's your typical standard curve, because you should be running your standard curve every time, right, and getting this data similar to what we get when we run the standards in our shop. And flow cytometry, you'll see histograms, you'll see dot plots, right, for the detection of specific antibodies, where in this case you have CD3 on one side and then CD8 on the other axis. Comparative to our isotype control, we don't see any staining, and we see great staining for CD8. We also are validating for our IHC and ICC applications. These are 2 examples on FFPE cells, a mouse example on the left, and then a human tissue example for IHC on the right. We also do ICCIF in terms of live cells either grown on cover slips or in chamber slides that can then be fixed and permed via a variety of different techniques. And recently we've added in some more multiplex immunofluorescence images and services that allow for this type of data to be collected. But with this said, we really do our best to validate and feel confident about the applications that we're putting out there for you, but we can't obviously test every single experimental condition. So with this being said, we are kind of all responsible for validating our antibodies. So when we think about these things, we're trying to do our best in terms of giving clear communication and messaging that's providing experimental data, results, as much data as we possibly can, right, and the applications that we've actually tested for. But you also need to have a clear understanding of what you're trying to use the antibodies for, right, what applications, is it validated for those, and at the same time be prepared to optimize our antibodies for your specific experimental conditions. We're giving you a range and we're hoping that it's going to work, but sometimes it may take a little bit of troubleshooting. And with that said, don't be hesitant to reach out to technical support, right? Have these conversations with us, because we are scientists in-house validating these. We're going to work with you to make sure that you can get it to work in your system, or that we've actually tested it in our system. And the kind of last points that we want to to leave you with is that really keep an eye out for some of the red flags when you're purchasing and evaluating antibodies on the market. Whether they're using only recombinant proteins or overexpressing cell lines. Sometimes that's the only option, right? And we have to take that with a grain of salt. Maybe they're not, folding in the appropriate ways. So instead, try to look for things where we do have biological expression of the target. Again, antibodies validated for only a single application. Again, sometimes these antibodies are really great and they work awesome in these applications, but when you can see the same data or similar data with multiple applications, you're just going to feel more confident that it's actually binding to the thing that you're trying to study. And lastly, single images of validation data, right? If it's validated for 7 applications, but there's only one piece of data, where's the rest of the data, right? You can reach out to us if we don't have it and we'll get it to you, right? But just be conscious of why is that data not there. So with that, we will leave you with our booth, and I'm doing a poster tomorrow on epitopes expression for multiplex assays, and we'll take any questions that you might have. So what happens if you buy an antibody, says it's going to work, and it doesn't? Brian's our head of technical support, but usually we'll try to work with you to figure out why it's not working. Is it actually validated for the application? What conditions are you using? Are they the same as the conditions we're using? We may send another sample to you to try a new lot or something else to really make sure that we're doing what we can to give you a quality product.
Yeah, and that really comes down to, I think, an application-specific type of approach with those antibodies. And so in circumstances where we have a cell line where we can look at mass spec data or transcript data and say we know that that antibody or that protein should not be expressed in that cell line, we will always bake those into our assays to make sure that we have a reliable negative control there to ensure that we're not seeing off-target stuff. If we have a protein target and we know that there are some very closely related proteins to that specific target, we will make overexpressing cell lines to ensure that we're not seeing binding to those targets and only to our target protein. If we have tissue that we know should be not staining positive on a given antibody, we're going to include that in our tissue array that we do during our IHC validation steps to ensure that we're not seeing staining in certain types of tissues while seeing staining in the ones that should be showing as staining. So wherever we practically can include negative data, we always do that. We don't always show those negative results in our data sheet because it's negative, so in theory you shouldn't be seeing anything, but wherever we can, we include negatives every time.
So she was asking if an antibody is validated for an application, say Western blot, and she runs it in flow cytometry. Is it guaranteed to work in flow cytometry? Will it work in flow cytometry? And under some conditions it will, right? I do our flow cytometry, I'm working through our catalog, and we haven't tested all of the antibodies for flow cytometry yet. Like, we're working on it, so if it doesn't have the flow cytometry application, it doesn't mean it's not gonna work, it may mean we just haven't tested it yet. But there are some conditions where I've tested antibodies that have been validated for IHC, Western blot, IP, ICC, and they don't work for flow cytometry. So it's not all go this way or all go this way. It's really application specific, which is why we do our best to make sure if it's on the data sheet, we have tested it in-house for that specific application under the conditions that we describe.